Welcome to part 2 of your transcription and translation help video. So this video is on translation, all right? So in the last video, transcription, we were solving the first 3 rows, all right? How to solve for the DNA row, how to solve the messenger RNA row, and the tRNA row. So in this video, we will be looking at how to solve the very last row, AA, which stands for amino acid, all right? And as you should already know, the amino acids are the building blocks of proteins. All right, so once we get these 9 sequences all figured out, these 9 amino acids together makes a protein. All right, oops, there we go. All right, let's get into it. Okay, so looking at the problem that I completed first. So in the first column, all right, so you see here I have MET, all right? MET stands for methionine, all right? And I have a codon chart that I gave to you guys in Google Classroom, and it looks like this right here. So if you want to print it out, then you can. I think I gave my face-to-face people a copy, all right? If not, then I'm sorry, I forgot to give you a copy. But if not, then you can just use the copy that I have in Google Classroom. All right, so before we even start trying to talk about amino acids and everything like that, the first thing that we have to remember is what we're going to be working with. All right, so amino acids, in order to figure out what the amino acids are, you must use a codon chart. All right, you must use the codon chart. Now, the important thing I want to point out about the codon chart is this right here. This is from your notes. You do not need to memorize this chart. You just not have to know how to actually read the chart. All right, so whenever you're going from messenger RNA, all right, so whenever you have finally completed your entire messenger RNA, then you can figure out your amino acid sequence, all right? Or other way around, if you have your amino acids sequence, then you can figure out what your messenger RNA is, all right? The reason why these two are connected is because the messenger RNA is like the actual language. All right, so imagine if someone is talking. All right, this is the person that's speaking. This is the translator that's letting you know what he is saying. All right, so that's why they are connected. In translation, the messenger RNA is what is actually being translated into these amino acids. Okay, the only reason why we figure out the tRNA is because you're just trying to see what each amino acid actually stands for in codon language. All right, if I was trying to give methionine its own codon language, then tRNA would be that. All right, but don't worry about tRNA. All right, don't worry about tRNA. You're only going to be looking at messenger RNA to figure out the amino acid. All right, so first let's look at how to read a codon chart. All right, so here's the codon chart that I gave you. So as you can see on this codon chart, it is separated into 1, 2, 3, 4, is 12 boxes. All right, 12 boxes in the middle. All right, and on the edges of the box, you see here first base in the codon, second base in the codon, and then third base in the codon. All right, so each of these sides are going to correlate to your messenger RNA. All right, so I'm just going to, there's no need to make up a messenger RNA because I have some right here already on our problem. All right, so just for the sake of the explanation of this video, to keep it simple, I'm going to write it on this paper. All right, so my first messenger RNA is AUG. All right, so this is the first one that I figured out on my problem, okay? But remember, in order to get AUG, I had to use my DNA first. All right, so again, depending on what number you're on the worksheet, you have to figure out your messenger RNA strand completely before you do the amino acid sequence. All right, so AUG. All right, so here's my first base, A. So looking at this codon chart, I'm only going to focus on the A's. All right, so here it is. AUG is right here. Here's my A, All right, so now I'm looking at this third row only. All right, since that's the first base in my codon. in my messenger RNA. All right, that's what I'm writing down here. All right, second base is a U, so I'm gonna follow the row. So I'm looking at this third row. Here's my second base, U, right here. So following along with the pattern, so looking at the third row, now I'm looking at this first block inside of the third row. All right, so I'm just focusing on this block right here. All right, so now in order to figure out which amino acid, which of these 4 amino acids are going to be the correct amino acid, the last letter G, here it is, third base in the codon, so following along with the box. So here it is, the G. If I follow the line, it's methionine, M-E-T. All right, so that is what I have written in my amino acid space, M-E-T. All right, you're only going to write the first 3 letters of each amino acid. You don't have to write the entire word out. This is right here. This is from the notes. All right, the entire word is, here it is, methionine is, you know, spelled out, and you don't have to worry about writing the entire word out. You're just doing the first 3 letters. All right, I'm just showing you this. This is from the notes. Just showing you what the entire word is. But the same thing, all right? Use an A, second letter U, third letter G. And there it is, methionine. All right, so that's one way to read a codon chart. Another way to read a codon chart is, let me show you this right here. All right, so all right, so there are some codon charts that you can find. You can pull it up on Google, that instead of being in a table format, it's in this sort of circular format, as you can see right here. Now, these are also perfectly acceptable to use, and some students even find these easier to read, but you know, it's up to you which one you want to use. So again, I'm using that same AUG from my messenger RNA, and I'm going to look at this codon chart. What is the amino acid? So this inner circle right here is going to be my first letter in the codon. So here's the A. My second letter is going to be this middle circle. All right, and then here's my U. So A, following the pattern, U. And then the last circle has G. There you go. So A, U, G, just following the line. There you have it, methionine again. So same codon chart, different shape. So either one of these will give you the same answer. All right, so let's look back at our problem. Okay, so I have a lot of my messenger RNA already solved. So I can use my messenger RNA right away to figure out my mRNA. All right, and so since these two are connected, I'm going to write my amino acid in green again. Okay, now some of your problems, you don't have your entire messenger RNA solved already. Some problems give you the DNA first. So like number 1, in that scenario, you need to use the DNA to figure out your messenger RNA. All right, some of the problems will give you the tRNA first. So you need to use the tRNA to figure out the messenger RNA. And then some will give you just amino acids. It'll give you the amino acid chart first. So in that scenario, I'll show you in a minute how to use this codon chart in reverse. All right, so but first let's use it the normal way. All right, so let's do these first 3 codons right here. So here I have my messenger RNA is GAG. So using GAG, I can figure out the amino acid. All right, so I'll use both codon charts. All right, so first base was G right here. Second base, A over here. So now G, I'm looking at this entire row down here. Second letter A, so now I'm looking at the third block in the last row. So G, A, and the last letter is G. Here it is. So that is going to be GLU. All right, and GLU stands for glutamine. All right, GLU. All right, so, all right, there's my amino acid. All right, next one, UG. G, and A. All right, so here's my codon chart. U, first letter, sorry, right here, U. So I'm going to focus on this row up here. Second base is a G. All right, so now I'm just looking at this block right here. And then A, UGA. Here's my A. Oh, hold on, that says UGA, that's a stop. Oops, sorry, it's not A, it's a U, UGU. Right here, CYS. All right, so my U, first base, and the second base was G, third base was a U. All right, so that's gonna be C, Y, and S. All right, and then for the last one, for number 4, I'll just use my circular codon chart just so you can see how it's the, might be a little bit simpler to read. All right, so here's my messenger RNA, GAG. So here's my circular codon chart. So here's my G, here's my A, and then right above it, here is my U. GAU, that is aspartic acid. So those first 3 letters is ASP. Oops, sorry, my camera. There we go. Okay, ASP, and it's the same thing over here as well. G down here, this entire row down here, this fourth row is where my first G's are. Second letter is was an A. Now I'm just looking at this third box, and then my last letter, here you go, was a U. Aspartic acid, ASP. All right, so that's how you do it the normal way. All right, going forwards. Now going backwards, if you get amino acids first, like this problem has in column number 5 and in number 8, it gave you the amino acid first. All right, so how do I figure out what the messenger RNA is with just using my amino acid? All right, so if you look at your codon chart, ALA, let me look at this codon chart right here. So if you look at the codon chart for ALA, look at right here. There are 1, 2, 3, 4 ALAs in this problem right here. All right, so just like it says in the directions at the very top of this worksheet, it's going to be like your choice. Is pretty much up to you what you want your code to be, what you want your messenger RNA to be. So it's up to you when you have multiple options like that. So for ALA, I can either, so here we go. This little box right here has all of the ALAs that I see. So I'm just going to write down all of the messenger RNAs that match with this box right here. All right, so the first one, G, C, U, G, C, C, G, C and A, G, C, G. All right, so as you can see, the first 2 letters are going to stay the same, all right, because I'm in the last row, which belongs to G, first base, G, second column, second base is C. So G and C is going to stay the same. But this last letter, this is the one where it's up to you. So you can either put for the messenger RNA, this one, any of these 4, all right? Either one is going to give you that exact same amino acid, all right? So I'm just going to use the first one, GCU, and that is my messenger RNA, GCU, all right? And this is the only time that it will be like viewer's choice when you have just the amino acid, all right? If it gives you any of the others, like tRNA or DNA, then you can figure out what the real messenger RNA is. But in this case, you can't figure it out, all right? So let's look at column number 8, which is proline. PRO stands for proline. So looking at my codon chart here, just like the other one, there are multiple prolines, okay? So you have 1, 2, 3, 4 different prolines on this one. So again, viewer's choice, all right? So, oops. All All right, so these prolines are located on this second row. So this second row belongs to first base is a C. So I'll write down here all of the options. So I'm gonna have, I might as well just do all 4 C's. Second letter in the base is a C again because it falls underneath this C. So again, all C. There we go. And then my last base, it can be either of the other letters, just like in the first one that we did. So it can be CCU, CCC, CCA, or CCG. All right. So again, just to keep it simple, doing the first one, CCU. All right, so my messenger RNA for this proline is C, C, and U. All right, but like I said, any of those options can go with it. All right, so now looky here. My entire messenger RNA is completed. All right, I was able to figure out the messenger RNA using what the problem was able to give me. So it's pretty much smooth sailing from here. I can figure out the rest of this easily because I have my key. I have my messenger RNA. So let me complete my DNA. All right, so column number 5, G, C, and U. This is transcription happening here. G is going to pair with my C. C is going to pair with the G. And the U with an A. All right, down here, column number 8, CCU, 2 G's, and the U pair with an A again. All right, now my DNA is done. So, completed the DNA row, completed the messenger RNA. Now I can do my tRNA. It doesn't really matter the order you go in. Once you have your messenger RNA, you can do the tRNA next or the amino acid. Either, it doesn't matter the order at that point. All right, so transcription again. So I have GCU in that messenger RNA. So C, G, A. And then I have CCU, G, G, A. All right, and now my transfer RNA is complete as well. All right. And now I have 3 amino acids I need to figure out, these last 3 amino acids. All right, so here is my messenger RNA, UAC. I'm going to use my circular chart right here. So here is my first letter, U, second letter, A, third letter, C. UAC is triosine, so that's just TYR. AAC, first letter A, second letter A, third letter C is asparagine. All right, not sure if I said that right, but ASP basically. All right, and then my last one, UUA. So let's look at the other codon chart for this last one. So UUA. So here's my first letter, U, focusing on this top row. All right, second letter, U. So now I'm just looking at this box right here that's in the top row. And the last letter, I'm sorry, not UUA, it was UAA. All right, so not that U. So let's start over. First letter, U, right here, looking at this row at the top. Second letter, A, looking at this box right here, second letter A. So now I'm just focusing on these 4. All right, and then my third letter is an A. All right, UUA is going to lead to a stop signal. All right, and this is just a signal that lets the ribosome know to stop making the protein. All right, MET is your universal start codon, as you can see in the notes right here. Methionine, universal start, that means start making the protein here. Stop is to stop. All right, let me zoom out a little bit so you can get a nice picture of all of this. And that is how you solve number 5, transcription and translation. Hopefully these videos were helpful to you. All right, hopefully you were able to use these to help you figure out numbers 1 through 4. If you have any questions for me, then please reach out to me, email me, and you can also always join my office hours tomorrow. All right, you guys have a great weekend. I will see you all on Monday.